विकत आणायला काय म्हणा तुला झाले कॉलेजचेच बुक तुझे तू वापरायला लागले.
गुड मॉर्निंग सर.
व्हेरी गुड मॉर्निंग सर.
हॅलो, आलं कोणी? बरोबर, मी मॅडमला फोन सांगतो. एक आमचं FDP चालू आहे. असं तिकडे पाठवलं चालेल, डिपार्टमेंटकडे पाठवा डायरेक्ट. हां हां, हो चालेल, तसं खूप चांगलं. मॅडम 2 ऍडमिशन तिकडे पाठवा. बाकी काय म्हणताय सर, काय चाललंय? सरांचं काय चाललंय अजून? सर आवाज येत नाही तुमचा.
गुड मॉर्निंग अंगरगे सर.
हा, व्हेरी गुड मॉर्निंग सर.
कसे
आवाज
आहेत
येत
सर?
नाही का?
हो येतोय सर. कसे आहेत अंगरगे सर?
मजेत. बरोबर. कुठे घरी की ऑफिस मध्ये?
नाही नाही, मी हाफ डे घेतला सर आज. दुपारून
बरोबर,
जाणार
हाफ डे
ऑफिसला.
घेतला.
हो सर.
चालेल
कारण
चालेल.
मग परत या परत जा ऑफिसला, लांब होतं.
हो हो हो,
हो, त्यामुळे मी दुपारून जाईल ऑफिसला सर.
चालतं चालतं.
हो, काय चाललं
आपण
सर, कसे आहेत?
साधारण एक 5 मिनिटांनी सुरू करू आपण.
काहीच
ओके.
प्रॉब्लेम नाही सर, काहीच प्रॉब्लेम.
आणि 12 12:15 पर्यंत संपू.
हो
हो की
चालेल
नाही?
सर. हो
हं?
हो चालेल सर, काहीच प्रॉब्लेम नाही.
बाकी काय म्हणते ड्रग डिस्कवरी?
चालू आहे सर. आता
चालेल.
थोडंसं वॅक्सिनेशनचं प्रोग्राम हे झाल्यामुळं चांगलं आहे आता.
बरोबर,
हो
चांगलं आहे.
हो. आणि सध्या काही लीडिंग कंपाऊंड्स पुढे चाललेत फेज ट्रायल्समध्ये. काहींनी क्लिनिकल ट्रायल्स कंप्लीट केल्या. तर
बरोबर. छान
चांगलं
छान,
आहे.
एकदम.
हो.
ओके, व्हेरी गुड. आठवण येते की नाही मग साहेबांची? काही लोक जॉईन होत आहेत अजून, त्याच्यामुळे थोडं थांबलो.
बरं बरं, काही प्रॉब्लेम नाही, काहीच प्रॉब्लेम.
हा हा, थोडं पाणी वगैरे घ्या. आता काही
हो,
इथं असेल तर दिलं असतं आम्ही तुम्हाला.
नाही काही प्रॉब्लेम नाही सर. थँक्यू सो मच.
नाही ना मॅडम. आमची पूर्ण फॅकल्टी आहे. फॅकल्टी डेव्हलपमेंट प्रोग्राम आहे ना सगळं, पूर्ण
हो हो,
फॅकल्टी.
ड्रग केमिस्ट्रीचं फॅकल्टी डेव्हलपमेंट प्रोग्राम सर.
हो हो, ड्रग केमिस्ट्री या वर्षीपासून सुरू करतोय आम्ही इकडं.
फार चांगले सर, फार चांगले.
हा,
कारण मुलांना नवीन नवीन संधी, नवीन नवीन गोष्टी माहिती पाहिजे सर.
बरोबर बरोबर. आम्हाला तुमचं इनपुट आता पाहिजे याच्यात.
मी कव्हर करतो सर बऱ्यापैकी.
हा
इवन
हा,
दो रोल ऑफ इंडियन फार्मास्युटिकल कंपनी पण कव्हर करणार आहे सर.
बरोबर.
ऍडिशनल थोडंसं
हा
ब्रीफ मध्ये
आता
कव्हर करल, पण
हा
मुलांना थोडीशी ब्रीफ आयडिया असेल असं कव्हर करणार.
हा हा, ठीक आहे. आता हे जे आहे ना सगळं फॅकल्टीसाठी आहे.
हो
म्हणजे फॅकल्टी आधी ट्रेंड झाले की खाली मुलांसाठी पण चांगलं होईल असं
हो
या
सर. हो
दृष्टीतून आता. तर जॉब ऑपॉर्च्युनिटीज वगैरे थोडंसं त्याच्यावर पण हायलाईट करता आलं तर करा.
चालेल सर, काहीच
जॉब
प्रॉब्लेम नाही.
ऑपॉर्च्युनिटीज.
हो.
शेख सर नमस्कार.
नमस्कार सर.
भिशे सर बोलतोय.
हो सर, नमस्कार. व्हेरी गुड मॉर्निंग
नमस्कार
सर.
नमस्कार. आ
व्हेरी गुड मॉर्निंग सर.
व्हेरी गुड मॉर्निंग. काय चाललंय?
बस सर, ठीक आहे.
काही पाऊस वगैरे आहे का तुमच्याकडे?
काल भरपूर पाऊस झाला सर, 2 तास
अरे
संध्याकाळी.
बापरे. होय का?
म्हणजे आता ह्या मोसम मधला सर्वात मोठा पाऊस होता सर.
अरे गुड गुड गुड.
हो सर.
मग आता संपत येता तुम्हाला दिलाय गुड न्यूज त्यांनी
हो
पावसाने.
सर. हो सर, कारण एवढ्या 1-1.5 महिन्यात मराठवाड्यात पाऊसच नाही सर जसा पाहिजे तसा.
नाही नाही, पाऊस नव्हताच 1.5 महिना झाला.
हो.
मराठवाड्यात
हो
पाऊसच
सर.
नाहीये.
हो सर.
त्याच्यामुळे हा एक पाऊस तरी चांगला, एक दिलासादायक आहे असं म्हणायला काय हरकत नाही.
हो सर, बरोबर.
हम्म. बाकी आपण आता चालू करणार आहोत या वर्षापासून.
हां.
थोडं MSc ची ज्या ठिकाणी आहे तिथून आपल्याला थोडीशी हेल्प हवी आहे, तर ती हेल्प आपण
हो.
बघूया.
हो सर, चालेल.
आणि तुमच्यासारखे लोक आम्हाला गाईड करायला आहेत. तुमचं गायडन्स आम्हाला खूप महत्वाचं आहे आता.
ग्रेट प्लेजर सर. हो.
अंगारगे सर आमच्याकडे आलेले आहेत आणि
हो
ते
सर.
सुद्धा त्याच्यामध्ये पारंगत आहेत त्या विषयामध्ये चांगले.
हो सर. आणि
हम्म.
त्यांचा ड्रग डिस्कव्हरीचा प्युअर मोठा एक्सपिरियन्स आहे सर. आणि
हो हो
त्यांचं
हो नाही आणि त्यांचा खूप मोठा एक्सपिरियन्स आहे.
हो
एवढंच होतं की जरा ते थोडे अगोदर आले असते म्हणजे आम्हाला अजून त्यांचा खूप फायदा झाला असता.
हो हो. आणि त्यांचं बेल्जियमचं पीडीएफ असल्यामुळं तिकडचं पण इनपुट घेता येईल सर. कारण ते
होय होय होय
आम्ही
निश्चित
मी
निश्चित
आणि
निश्चित.
हंगरगे सर बरोबरच होतो. तर
होय होय
त्यांचा
होय
बाहेरचा एक्सपिरियन्स पण फार मोठा आहे सर.
नाही आणि त्यांच्याच आता त्यांच्याच अंडर सुपरविजन सगळं चाललेलं आहे म्हणायचं थोडक्यात आता.
हो सर. आणि न्यू जनरेशनला ड्रग केमिस्ट्री म्हणजे एक नवीन अपॉर्च्युनिटी आहे सर.
होय 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 बरोबर आहे बरोबर आहे.
सध्याच्या ह्या याच्यात आहे ना सर,
हम्म.
असे व्हायरल इन्फेक्शनचे आजार येत आहेत वेगवेगळे. आणि त्याच्यासाठी नवीन म्हणजे ड्रग डिस्कव्हरी तर एकदम कंपल्सरी झाली सर आता. ड्रग
नाही
डेव्हलपमेंट,
नाही
ड्रग
सर
केमिस्ट्री.
याला तुम्हाला ड्रगला कुठे तुम्हाला कधी आपण जसे म्हणतो ना सलूनचे दुकान आहेत, चपल्याची दुकानं
हो
आहेत, हे जसे
हो
बंद पडणार नाहीत तसं ड्रग बंदच पडणार नाही ना कुठे.
हो सर. बरोबर आहे, बरोबर आहे. आणि
ड्रग हा
व्हायरस पण
तुम्हाला कायम लागणारच आहे.
बरोबर आहे सर. आणि
आणि
एखादा व्हायरस हॅलो
हा सर बोला ना.
एखादा व्हायरस आला तर तो म्युटेट होत चालतो. अल्फा, बीटा, डेल्टा, गॅमा.
येस 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 बरोबर आहे सर.
ते एका ठिकाणी कंट्रोल राहत नाही सर. एकाच आजाराचे 10,000 लॉंग टर्म होऊन जातात.
होय 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 बरोबर आहे.
त्याच्यासाठी हा प्रोग्राम किंवा हा विषय हा म्हणजे डेव्हलप करणं किंवा मुलं याच्यात हे करणं फार आवश्यक झालंय सर.
आणि तसं आता स्किल सर नाहीत आहेत ना लोक, समजा स्किल लोक एवढी
हो.
मिळत ना झालेत ना आपल्याकडं.
नाही.
ड्रग हा विषयच नाही ना कुठे अंडर ग्रॅज्युएशनला.
बरोबर आहे सर, बरोबर.
म्हणजे
आणि सर
हा
हा सर बोला.
आपल्याकडे आता आपण संपूर्ण महाराष्ट्रात जर पाहिलं तर ड्रग विषय अंडर ग्रॅज्युएशनला असणारं आमचं एकमेव कॉलेज झालं असं मला वाटतंय.
हो सर. आणि अजून गोष्ट म्हणजे काय आहे का आपल्या देशात इन्व्हेन्शनला फार महत्व आहे सर. बाकीचे देश
होय होय होय
आपल्याकडचे
बरोबर आहे.
कॅन्डिडेट घेतात सर इन्व्हेंट केलेले. आणि आपले
हम्म.
मुलं तेवढे ऑनेस्ट आणि हुशार आणि
हम्म.
डेडिकेटेड असतात सर विषयाला आपल्या.
बरोबर आहे. आमच्या कॉलेजला
परदेशात
सर
हा.
CIII चालू केलेला आहे आम्ही, कॉलेज इन्व्हेन्शन इनोव्हेशन अँड इन्क्युबेशन.
अरे वा वा. फार
म्हणजे
छान आहे
ते
सर.
चालूच आहे आमच्याकडे.
हो हो. आता
त्याला
सर
आपण हा
परदेशात आहे ना सर, मॅक्सिमम इनोव्हेशनला भारतातलेच मुलं घेतात सर.
निश्चित
ते
निश्चित सर निश्चित.
हो.
आणि
अमेरिका झालं.
आपल्याकडे थोडंसं सर हार्ड वर्किंग सुद्धा मुलांच्या मध्ये टेंडन्सी आहे.
हो सर, हो सर.
आणि
आणि
टॅलेंट्स
मुल
ही आहे तशी मुलांच्या मध्ये.
हो सर. आणि मुलं फोकस्ड असतात सर. कष्ट करतात, त्यांच्याकडे टॅलेंट
हा
असतो आणि फोकस्ड करतात. त्यामुळे कुठं ना कुठं चांगलं इनपुट मिळतं सर.
बरोबर आहे. निश्चित निश्चित अगदी निश्चित चांगलं इनपुट मिळतं आपल्याला.
हो
आणि ह्या कोर्स सुद्धा आम्हाला चांगलं इनपुट मिळेल. फक्त त्याला जरा
हो
फळं यायला थोडेसे वेळ लागेल, पण चांगली
हो.
त्याला फ्रुट्स येतील असं आमची अपेक्षा आहे.
बरोबर.
आणि तुमच्या सारख्यांचं आम्हाला जर मार्गदर्शन मिळालं तर निश्चितच आपण याच्यामध्ये चांगलं काहीतरी करू शकतोय.
हो सर, निश्चितच सर.
हा खन्ना सर सुद्धा आम्हाला चांगलं गायडन्स करतायत.
हो सर. खन्ना सरांचं पण आम्ही आता तसं म्हणायला गेलो खन्ना सरांचे स्टुडंटच आहे सर आम्ही. मी श्रीरामपूरचा आहे सर
हा नाही
प्रॉपर.
नाही ते हा कुठले
श्रीरामपूर,
गाव कुठले?
श्रीरामपूर,
देवापूर
श्रीरामपूर,
श्रीरामपूरच्या अच्छा
अहमदनगर
अच्छा हा
जिल्हा.
हा
हो.
हा
माझं रयतमध्ये
नाही
पूर्ण
मला माहित
शिक्षण
आहे.
झालंय सर. हो.
ते कुमावत म्हणून माझ्या बरोबर बॉटनीचे एक होते बेलापूरचे.
हो, हो. हो, हो, हो. डॉक्टर कुमावत सर माहिती आहे मला सर.
हा ते माझे चांगले मित्र आहेत सर.
हो, हो.
आता ते
रयत शिक्षण संस्था.
हा रयतमध्येच आहेत, पुण्याला आहेत आता ते.
हो, 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 हो. बरोबर.
बरोबर आहे.
श्रीरामपूरला RBNB कॉलेज आहे. तिथून माझं MSc शिक्षण झालं सर.
गुड गुड गुड. अहो सर मी ते
रयत
तुमचं
शिक्षण
हायस्कूल
संस्था.
आहे ना रयतचं,
हो.
त्या रयतचा विद्यार्थी आहे मी हायस्कूलचा त्या.
अरे अरे सर, अरे वा. कारण आमच्या नगर जिल्ह्यात रयतचं फार मोठं हे आहे सर. शाळा,
जाळ आहे
कॉलेजेस.
पूर्ण.
हो.
आमच्या साताऱ्यानंतर नगर जिल्हा फार मोठा रयतनं व्यापलेला आहे सगळा.
हो सर, हो सर.
बाकी अजून काय म्हणताय?
काही नाही सर. 20 वर्षापासून ड्रग डिस्कव्हरीत आहे सर.
व्हेरी गुड सर.
आणि 17 वर्षापासून ओखाड R&D लाच आहे ड्रग डिस्कव्हरीमध्ये.
गुड.
मॅक्सिमम एक्सपिरियन्स हा ड्रग डिस्कव्हरीचा आहे आणि मी
अच्छा
पण
अच्छा.
छोट्या गावातून आलेलो आहे सर. आणि ह्या
हा.
डिपार्टमेंटमध्ये म्हणजे तेव्हा ड्रग डिस्कव्हरी बिगिनिंग स्टेजला होती सर. पण आता
नाही, बरोबर आहे, बरोबर आहे, बरोबर आहे.
जगाच्या पाठीवर भारताचं पण चांगलं नाव असल्यामुळं वेगवेगळ्या कंपन्या, वेगवेगळे रिसर्च फंडिंग येत आहेत.
हा.
त्यामुळं फार चांगलं आहे सर. चांगले चांगले
निश्चित
इन्स्टिट्यूट
निश्चित.
मधून चांगले मुलं येत आहेत.
हम्म.
आता हंगरगे सर पण बेल्जियमला होते ना सर, तर त्यांची
हा.
इन्स्टिट्यूट फार मोठी होती. त्यांचा एक्सपिरियन्स पण फार ह्यूज आहे सर. कारण
नाही
आम्ही
नाही,
बरोबर काम
लुपिनचं.
केलेलं. हो.
हा, लुपिनचं मोठंच आहे ना सर.
हो, लुपिनचं आणि वोखाडला पण होते ते. त्याच्या आधी बेल्जियमहून आले होते ना सर, तर
हा
त्यांचा
हा.
बेल्जियमचा रिसर्च प्रोग्राम पण मोठा होता.
होय होय, नाही
तुम्हाला
ते आहे
निश्चितच
त्यांचं.
फार मोठी हेल्प होईल सर त्याची.
हा, नाही ते व्यक्तिमत्वच मोठं आहे हंगारगे
हो
सर.
सर. हो सर. हो.
नाही, आम्हाला त्यांचा खूप उपयोग होणार आहे याच्यामध्ये.
हो.
भरण मॅडम.
Sir, yes, sir.
हॅलो.
Yes, sir.
चालू करताय का?
हो sir, yes.
चालू करा.
Good morning and warm welcome to the Honorable B.T. Jadhav sir, YCI Satara, Mr. P.V. Bise sir, Head Department of Chemistry, BOS Chairman Dr. V.V. Sawant ma'am, Drug Chemistry Coordinator Dr. P.A. Bharad ma'am, and all members for the discipline specific program 2021-22 on syllabus drug chemistry diploma and skill course. Now I request to Mr. P.V. Bisesar, Head Department of Chemistry, to welcome his resource. To you, sir.
Okay. Good morning to all. Am I audible to you?
Yes sir.
Yes. Sir, the Department of Chemistry today have organized the FDP program in the drug chemistry. Actually, sir, this is our 4th day that we have organized the FDP program, particularly in the subject of chemistry, first year chemistry, then diploma, advanced diploma, then MSc first year, and skill courses. So this is, I think, 4th day of the FDP program. Sir, our college is the autonomous college running UG and PG in different subjects. Under the roof of Department of Chemistry, we have run this drug chemistry as one of the entire subject at a BSc level. We also run MSc in 4 different branches, that is analytical, physical, inorganic, and organic chemistry. And the intake capacity of the MSc is 176, sir. So this is the largest department as far as our college is concerned. So along with this traditional UG, PG, and research center, we run the diploma and advanced diploma courses for the different classes. We also run the skill courses for the different classes. And along with that, we have add-on course, capsule course. And now we have started under the roof of chemistry, drug chemistry entire as a one of the subject at a BSc level, sir. So this is the unique, I think, in the Maharashtra. We are the first to begin or to start this drug chemistry as an entire course at BSc level. And I know that this course or this subject at the BSc level will be helpful for the students for their betterment of the future. When I have discussed with the Khanna sir that if any faculty we want, do you provide us the faculty? And he says that now there is no any faculty, because whatever the student passes from our college at MSc, they all get the jobs, and there is 100% placement of the students who have passed MSc in the drug chemistry. So it is a better thing, and it is a very good thing that the students who have passed the MSc in the drug chemistry has got the 100% placement. And in the similar way, we also expect the same thing from the students who have passed their graduation by taking a drug chemistry as a subject. Now, as far as the FDP program is concerned, what this FDP program is organized only for the teachers who will teach to the students of the drug chemistry. And our expectation from the expert is that up to how how much depth you should enter in the subject. So that is the main expectation of the FDP program. And the persons like you, sir, Sheikh sir, you are
Okay.
you have a great experience. You have large experience in the drug discovery, and you are working at one of the most special institute at Aurangabad. Similarly, one more person who is a resource person of today's FDP program, that is Hangarge sir. He has also great experience in the field of drug. So both you will guide us, and we tell us that is up to what depth we should enter in the subject. So the FDP program which we have organized for the teachers will be helpful to learn something from your side, and it will have a grand success, and in a whole day we will be with a drug chemistry. So once again I welcome you, sir, Sheikh sir, and Hangarge sir, for this FDP program. And I also thanks to the organizer that they have organized this FDP program, particularly in the drug chemistry. So thank you very much.
Thank you,
Over
sir.
to anchor
Thank you,
Vinaya
sir.
Thank you,
ma'am.
sir.
Thank you, sir. Now I request Dr. P. A. Bharat Madam to introduce today's speaker, Dr. M. U. Sheikh, Group Leader, Okhad Research Center, Aurangabad. Over to you, ma'am.
Good morning, all of you. First of all, I welcome Dr. M.U. Sheikh sir in this faculty development program, and it's my privilege to introduce our chief guest, Dr. M.U. Sheikh sir. Sheikh sir is a group leader at Okhad Research Center, Aurangabad at present. Sir has completed his master's from RBMB College, Sri Rampur in organic chemistry. Sir has done his PhD from Department of Chemistry, Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad. Sir has 5 patents and 12 papers to his credit. Sir has presented paper in many national and international conferences and workshops. Sir has 18 years experience in new drug discovery at different places. Sir has worked Herman Phenochem Limited and Suven Life Sciences Limited previously. With this brief introduction, I request Dr. Sheikh sir to guide us on drug chemistry. Over to you, sir.
Thank you so much, ma'am, for nice introduction. Shall I start, sir?
Yes, sir.
Yes, sir. Yes, sir. Yes, sir.
त्यांना स्क्रीन शेअर दिली का?
Very good morning to all of you.
Good morning, sir.
I am very much thankful to Professor P.V. Bise sir, Head of Department. I am also thankful Dr. B.T. Jadhav sir, Director Yashwantrao Chavan Institute of Science, Dr. V.V. Sawant, BOA Chairman, Dr. R.V. Hangarge sir, Assistant Professor, and Dr. Bharat Madam, Drug Chemistry Coordinator, for inviting me as a resource person for faculty development program in drug chemistry. Today, I am here to present novel drug discovery and development program. My presentation contains introduction to drug discovery and development, role of Indian pharmaceutical companies, pipelines of Indian companies, and conclusion. Then, what is drug? Drug is a chemical which is used as a medicine, simple definition. But the definition by Pharma company or pharma analogs is a drug, a chemical substance of known structure other than a nutrient or an essential dietary ingredient, which when administered to a living organism produces a biological effect. That is, drug is any substance or product that is used or intended to be used to modify or explore the psychological system or pathological state in the benefit of the recipient. Any substance or product that is used in the treatment, cure, prevention, or diagnosis of disease, or used to otherwise enhance physical or mental well-being. What is branded drug versus generic drugs? Branded drugs, which is known as innovator drugs. Branded drug is the product that is patented by innovator company. Branded drugs can be produced and sold by the company holding patents. Branded drugs are costly due to heavy R&D expenses. Generic drugs. Comparatively, generic drugs are cheap, cheap due to less development cost. Compared to branded drugs, the dosage form, strength, route and administration, quality, performance, and intended use. Generic drugs can be produced and sold after original patent expires. Brief drug discovery research program. Synthesis and biological screening of compounds primary take place, which will be entered into lead optimization stage. Then after lead optimization, the candidate development, that is chirality, salt formation, polymer formation. Then the drug enter into preclinical studies. And after preclinical studies, the compound enters into IND and clinical development program. IND means investigational new drug application. Current federal law requires that a drug be subject of an approval marketing before it transported or distributed across the state levels, but failures due to the non-drug like properties at preclinical stages. What is drug discovery program? Drug discovery is the process through which potential new medicines are identified. It involves a wide range of scientific disciplines, including biology, chemistry, and pharmacology. Drug discovery is the process by which new candidate medications are discovered. Once a compound that fulfills all these requirements has been identified, the process of drug development continues. There are three stages of new drug synthesis. Stage 1, drug discovery. Candidate molecules are chosen on the basis of their pharmacological properties. Say stage 2, preclinical development. Non-human studies are performed like toxicity testing, pharmacokinetic analysis, and formulation. Stage 3, clinical development. The selected compound is tested for efficacy, side effects, and potential dangers in volunteers and patients. Process of drug discovery. Stage drug discovery and development. Preclinical research, clinical research, FDA review, and marketing. These are the process of drug discovery. History. In brief, I will explain history of drug discovery. In pre-1990s, herbal drugs and serendipitous discoveries are there. In 1920s and 30s, vitamins and vaccine discoveries are there. In 1940s, antibiotic era is there and R&D boost due to World War II. In 1950s, new technology and discovery of DNA. In 1960s, breakthrough in etiology. In 1970s, rise of biotechnology and use of information technology. In 1980s, commercialization of drug discovery and combinatorial chemistry. In 1990s, robotic and atomization take place. Total drug discovery research program. Drug discovery, firstly 100 projects are taken in drug discovery program. In that program, target selection, lead finding, lead optimization of molecule, and pharmacologic profiling. It will take around 2 to 5 years. After that, preclinical development takes place. In preclinical development, pharmacokinetics, short-term toxicology effect, formulation, synthesis, and scale-up. It will take around 1.5 to 2 years. From 100 projects, 20 compounds were selected. And from drug discovery to preclinical stage, drug candidates should be selected. After that, clinical development take place. In clinical development, three phases are there: phase 1, phase 2, phase 3. Phase 1, pharmacokinetic study, tolerability, side effects in healthy volunteers. In phase 2, small scale trials in patients to assess efficacy and dosage. Long term toxicology studies were done. In phase 3, large scale controlled clinical trials will take. In between phase 1 to phase 3, around 5 to 7 years time, and 20 compounds, 2 drug candidates were identified. And after that, the compound enters in regulatory market for regulatory approval, submission of full data and review by regulatory agencies. And last and final stage is post marketing surveillance. Clinical development to regulatory approval, the regulatory submission is must. And from regulatory approval to post marketing surveillance, drug approved for marketing. The stages of development of a typical new drug, synthetic compound will develop for synthetic use. Why R&D costs are very high? Challenges for drug discovery. 10,000 drug candidates were selected for development. Drug candidates should be potent, selective, reversible, targeted, soluble, long-lasting, metabolically stable, permeable, and main and important stage is toxicity study. Drug candidates should be non-toxic, non-mutagenic, non-teratogenic, physically stable, patentable, and manufacturable. From 10,000 drug candidates, one molecule were developed, finding one molecule that meets a multiple criteria. Desirable profile of new chemical entity. Profile of new chemical entity. Integrity, API stability, log P, solubility, pharmacokinetic study, permeability, PYP 450 inhibition, metabolic stability, blood-brain barrier. Further test for new chemical entity to become drug. Drug tested in vitro, in vivo efficacy in animals, pharmacokinetic and pharmacodynamic studies, safety in animals, formulation studies, levels in healthy subjects, efficacy in patients. Ethnic group P3. Drug discovery, a multi-step process. Estimated international drug discovery cost around $300 to $500 million. Starting from new chemical entity synthesis, the compound enters in vitro and in vivo screening. That is lead identification of the molecules. After lead identification, the compound enters in preclinical toxicity study. That is preclinical toxicology. This is multidisciplinary discovery phase. It will take around 3 to 4 years and cost around 20%. After that, it will enter into clinical trials, IND filing. Clinical trials, phase 1, phase 2, phase 3. That is clinical research, and it will take around 3 to 6 years and cost around 55%. In between preclinical toxicology and clinical trials, applied research is there. The developed drug should be scalable, analytical testings, all analytical data developed, manufacturing, and dosage form. It will cost around 25%. After that, the compounds enter in FDA review of NDA, that is regulatory market. It will take around 2 years, and after that, the compound or drug enter into market. NDA is nothing but investigational new drug application, and IND is nothing but investigational new drug application, and NDA is new drug application. That is from clinical trial stage to FDA, NDA is required. Reason why drugs get failure, reason for failure of drug. There are 5% commercial reasons for drug failure, animal toxicity around 11%, adverse effect in human around 10%, lack of efficacy around 30%, poor pharmacological properties around 39%. In brief, I will explain role of Indian pharmaceutical companies in drug discovery. Drug discovery research setups in India are complete drug discovery setups are computational chemistry, medicinal chemistry, analytical process, biological research, pharmacokinetic, pharmacodynamic studies, toxicology studies, clinical and regulatory market. It will having Okhad R&D Center, Dr. Reddy's Lab, Ranbaxy, Zydus Cadila, Glenmark, and Orchid having complete drug discovery setups. And CRO, custom research organizations are computational, Schrodinger, medicinal chemistry, Sci Adventinium, analytical Vimta labs, process Almilo, biology and toxicology, Advinas, clinical Sigma labs. And almost many companies are entered nowadays in CRO in India. Business models in India. Developed drug on its own risk. Okhad and Zydus had developed drugs on its own risk. Development of NC to market level, total risk is taken by company. Then collaborate with MNCs. Glenmark, Dr. Reddy's and Orchid have collaborate with multinational companies. Development of new chemical entity to the preclinical or phase 1 and collaborate with multinational company for further development. Risk sharing with multinational companies. Part knowledge based services providers are confined to developmental services as required. Sci Adventinium, Advinas, GVK Life Sciences, and Vimta Labs. Scenario in drug discovery program. Work in already established therapeutic area as development guidelines and liabilities are known. Mostly work on weaknesses and advantages over competitor drugs in the market for the product to be superior somewhere spectrum toxicity oral OD. Classical structural activity relationship development guided often by computational means, mostly dependent on quicker in vitro test to screen compounds. Compromises on failurely decisions due to pressure to perform. Less culture of interdisciplinary research. These are some drug molecules of Zydus Cadila in different therapeutic areas and which are under development stage in phase 1, 2, and 3 trials. These are some drug molecules developed by Ranbaxy and with different therapeutic area and in different stages. Dr. Reddy's lab having 6-7 compounds under development stage. Glenmark having 7 compounds under development stage in different. Lupin having anti-TB, anti-diabetics, some of other disease in different developing stage. Piramal Healthcare also having different category and different stage of developments. Okhad having 4 molecules, 2 molecules were launched in Indian market as MROC and MROC-O, and 2 compounds in phase 3 clinical trials in USA. Conclusion: drug discovery process is tedious and expensive, where the risk of failure is more. Most of the Indian origins drugs are herbal medicines, where research expenses are less compared to the synthetic drugs. Need to train discovery people from beginning if results are required. Need to develop interdisciplinary teams, and training also required for an individual to become a team player. Next 10 years are crucial for Indian drug discovery researchers, as many molecules under final stages and development in many Indian companies will decide the path to go forward or backward. For a student, there are different departments in drug discovery. That is new chemical synthesis, that is chemistry department, where new chemical entities were synthesized. That compounds is goes to analytical department for their different characterization data, and after characterization the compounds goes to biological department, were tested in vivo and in vitro studies and take call for further studies. In new drug discovery, synthesis department is opportunity for organic chemistry students, and analytical chemistry students have requirement in analytical development labs. Also, there are toxicology, pharmacokinetics, microbiology, pharmacodynamics department, which is helpful for the drug chemistry students. As well as there are intellectual property research, IPR department, project management department, as well as drug regulatory department. This is opportunity for the new students. Also in formulation and development, also required for in biotechnology, as well as clinical research. Drug chemistry candidates are required, and in India and abroad, so many opportunities for drug chemistry students. Many institutes are right now, many institutes are containing research in drug chemistry, as well as many pharma companies in India, as well as multinational companies in India, gives opportunity to the drug chemistry students. Other than synthetic, many scientific writers, as well as chemical coordinators, many departments. I will share some slide to Dr. Hangarge sir, that gives many opportunities and some links for drug chemistry students. Thank you so much. Any questions? Thank you so much. Any questions?
Sir, I have.
Haan, sir.
Can you
sir.
go to your WCK file? WCK slide.
Slide ka, just a minute.
I'm interested in 787771.
Sir, you are work on this project, sir.
Yes,
Hangarge
I
sir.
know that, but I wanted to know about little bit
Okay.
over that WCK 771 and 787. Last button, I think.
Haan, sir. This one.
Yes.
Haan, sir.
So what is that, the 4873?
This is ketolide, sir.
Ketolide, okay.
Haan.
And 5222?
And this is ESBL inhibitor, sir.
Okay,
Beta
that is
lactase
new, right?
inhibitor. Yes.
So till 4873 I have worked.
You have worked, sir. Macrolide ketolide oxazorinone project,
Yes.
sir. Yes.
So what is the status of this one? It is in phase 3.
Almost 4873 almost completed, sir. And 5222 maybe in 6 months it may completed their phase 3 clinical trials.
Wow, excellent.
How, sir? And you are the part of 4873, sir. WCK 4873.
Yes, I was.
As well as WCK 771 and WCK 2349 also, sir.
WCK 771, Rajesh and you, Ganesh, these guys work on this, isn't it?
Yes, sir. And you are also help in metabolic study, sir.
Metabolites.
Metabolites in 2349 also, sir.
2, 3, 4. And WCK 771, it is a quinolone, no? Fluoroquinolone.
Fluoroquinolone, sir. Class of fluoroquinolone.
And its marketed name is Emroc.
Emroc 771 and MROC O 2349 right now in Indian market, sir.
Okay, in Indian. It is launched in the market, isn't it?
Launch lot already launched 1 year back, sir.
Very good. So my question is that can you little bit elaborate on the pharmacokinetics?
Pharmacokinetic studies, sir.
Yes, yes. So
Just
there
a
is
minute,
some absorption, distribution, metabolism, excretion. Some points are there, no? So in absorption,
Pharmacokinetics,
what
na?
are the criteria should be required in the drug?
Okay, sir. Just a minute, sir.
I think it is in your slide also somewhere.
Just, I will elaborate, sir. Pharmacokinetic studies, na, sir.
Yes, yes.
Just a minute.
So आवाज येत नाही सर.
सरांना अनम्यूट करायला सांगा.
Pharmacokinetics is defined as study of time courses of during absorption, distribution, metabolism, and excretion, sir. Clinical pharmacokinetics is the application of pharmacokinetic principles to the safe and effective therapeutic management of drugs in an individual patients.
Okay. So if you want to improve the PK profile, sometimes what happens, most of the drugs get failed in PK. Okay.
Yes.
At the early stage only, in the preclinical, most of the drugs get failed. And
Yes,
we know that the rule, fail fast. Okay.
Yes,
So if you want to improve that absorption of the particular drug, so what are the precautions or what are the measures we have to be considered?
sir.
To
Major
improve the
part
absorption of the drug, bioavailability.
oral viability, that is solubility is main factor, sir. Polar should compound should be in the salt formation or in the way polar group or penetrating group like carboxylic acids, sodium salt, or amino acid products that will penetrate and enhance their oral viability, sir.
Yes, yes.
Functional groups should be polar, sir, and water soluble, mostly water soluble, because in 10% DMSO compound get solidified and it will not enter in the cells, sir. That's why we will prefer to make it salts or amino acid products. If compounds create some problem in solubility, we will make it polar. That's why its oral viability enhances, sir, and it will release as a parent drug.
Okay, that's true. But if you are having salts, so salts are having more solubility, isn't it? But
Yes, sir.
Once that swallowed that drug, particular drug, then the excretion is faster than the normal drug, isn't it?
Yes, sir. Yes,
So
sir.
what means prodrug is better or the salt is better? Which one is better? What do you think?
Sir, most probability we will prefer false salt, sir. If salt creates some problems, having some excretion issues or some issues, then we will go for prodrug to release the original drug in the body.
Okay. Okay, good. Thank you, sir. And one more thing,
Yes,
what is the status of 3023 product or 2032?
Sir, frankly speaking, due to some toxicity issue, you are the you play key role in development of 3023 and 4086 drugs, sir. And you are the inventor of that compounds. But for after further studies, oxazolidinone class having some toxicity issue. That's why that project on hold, sir.
Okay, okay.
Okay.
Good. Thank you, sir.
Thank you so much, sir.
One moment, madam.
Thank you so much, sir, for your nice presentation. It will really help us when we will teach our student, and we will need your help in future also for many
Okay.
things. So we will contact you time to time. So I have a small question. What is expectation from like you are in the position holder in industry? So what is the expectation from students to join in industry as when they will be completing the BSc drug chemistry, like projects and all? How you expect they should complete their projects and up to
Uh,
which step?
okay. Um, I think so. Students should be familiar with some instruments like purification instrument like combi flash instrument, HPLC. Not only they have some training 1 month or industrial project. Yeah, industrial training should require for student for 1 month or 2 months. They were well familiar with instruments. Synthetically they are hardworking, all knowledge is there, but due to um, um, uh, they are not familiar with instrument, they are hesitating to interpret data or handling instrument. During the course, students should get 1 year, like MPhA student having industrial training 6 months in industry in final years. In our course also some training should be necessary from industry for students. They were comfortably do their jobs and they are selected anywhere after training because they are familiar with all the things which are going or currently latest instruments, latest technology, latest purification technologies. They are familiar with biological studies, biological instruments. I think so our students need 1 month or 2 months training program from industry. They were comfortably how to talk and face the different institute industry interviews and face all the problem because they are regularly familiar and observe all the instrument in 1 2 months and completed their training. They will go for further studies. I think so. The training should be required from industry. Students training 1 2 months required for industry.
Okay, thank you, sir. So, with this regard, I just want to ask, is industry allowed for this 1 month type of training?
Um,
If we,
um, uh, M-Farm courses they have compulsory to take course from
To work our
industry or
students in
certificate.
your
If
industries for 1 or 2 months like
uh,
this training program.
yes, sir.
Can we get a permission in your company like Vocard,
Uh,
the other industry like Harman Industries, Suven, any
uh,
laboratories?
uh,
Students get permission to work or carry out their project in that company
Sir, proper college or higher
or
authority
entrance.
can communicate with industry. They will definitely give permissions or industrial visit for few days. Hello, they will permit, sir, because right now some of the students having completed their graduation and joined somewhere, but after joining they know all the things and they will take for postgraduate admission because they know all the things, instrument, working culture, all the things. I think so if college or institute of properly communicate with industrial peoples or HR, they will give permission, at least industrial visit permissions they will give, sir. Some of
But I
the
think
yes,
every
sir.
industry have a compulsion. I think every
Huh.
industry have a compulsion to give a training to the students if the students are coming to them.
Yes, sir. Right now government also government also started last 2 years back, sir, some industrial training program giving stipend to students and completed
Yes,
their
yes.
training, sir.
That is what I am going to tell.
EPPF training program is there, sir.
Yes, 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 sir.
Yes,
Yes.
sir. They will definitely help. And sir, from last 2 years, Hangarge sir, in our labs, 8 to 10 students were completed their APPF training and got good opportunities in multinational companies, sir. Because they
Wow,
are
great.
familiar with all the instruments and technologies, sir. And 1, 2 students are in abroad, sir.
So even we have industrial chemistry as one of the entire course. And in that course also 1 month training is compulsory. So
Yes,
we
sir.
have started this program at Panvel, and nearby Panvel there are lot of industries.
Yes, sir.
So they have a compulsion. The industry have a compulsion that to take
Compulsion.
either 2 or 3 students at least
At least.
for the
Yes, sir.
industrial training.
Yes, sir. Correct, sir. Correct, sir.
Yes.
Because in regular studies, sir,
Yeah.
students are busy in their theory and practicals only. But
Oh,
students exactly instrument and data or interpretation of data take this knowledge from industry only, sir.
yes, yes.
All the
Until and unless
familiar.
they go the industries, they will not get the all the
I,
detailed knowledge of the working program.
correct, sir. Correct, sir.
If
Sir, we
people
learn
want to,
lot. We learn lot of things from Hungarge, sir, because they know from Belgium all the techniques, and they have
Yes,
entered purification technique in our lab, sir. That
yes.
automatic purification technique, Hungarge sir's idea is there, sir, because they have handled that
Oh,
instrument in Belgium,
that's nice. Nice.
and they introduce here. Hungarge sir, correct, na?
yes, that's
That's
good.
why it will help, sir. Somebody knows
That
what is the
that
thing.
that means we are very lucky, sir. That
Sir.
Hangargate sir is with us.
Really, sir. Really, sir.
Thank you, sir.
Okay. Thank you.
Sir, we have one more question. Shall we sign any MOU with WOCAD and YCIS?
Yes.
Uh, uh, yes, sir. Dr. Bhausar, sir, is vice president and your best
Okay,
friend, sir,
okay.
and your PhD colleague, sir. It is very easy to communicate to you, sir. Very easy, sir.
Okay.
क्योंकि
I
Bhausar,
will
sir.
talk with Dr. Sir.
Oh, sir. And it is possible, sir. Right now, that government allows these things to
Okay,
tie up
very good.
and these things. Sir, Bhausar, sir, having all chats, sir, you know, and your friend is my boss, sir.
Okay, okay.
Okay, okay,
So I will talk with him.
okay. Thank you.
Okay.
Thank you so much.
Thank you, sir, for such a nice information and guidance. Now I request to Dr. P. A. Bharad ma'am to express vote of thanks.
Thank you so much, Vinaya ma'am. It's my privilege to ask for this vote of thanks for this inaugural session. First of all, I thank our resource person, Muhammad Sheikh sir, for guiding us on drug chemistry in detail, as we are very new to this field, so it will really help us for future endorsement. Uh, I also thank our principal, Dr. B.T. Jadhav sir, and our head Department of Chemistry, Mr. P.V. Bise sir, for always supporting us. I also thank uh, BOS Chairman, Department of Chemistry, VVS Sawant ma'am, for joining this FDP. I also thank all senior faculty members for joining this FDP, and I also thank my colleague for patient listening this inaugural session. Thank you one and all. Over to the
Thank
anchor.
you. Thank you so much.
Thank you, sir.
Thank you, sir.
Thank you,
Have a
ma'am.
nice.
Inaugural session over. Session first will be start. Now I request Dr. P again P. A. Bharad madam to introduce to session first speaker, Dr. R. V. Hangarge, Assistant Professor, Department of Chemistry, YCS Satara. Over to you, ma'am.
आप कर रहे क्या टेंस कर रहे आप टेंस
Sorry. I'm very happy to introduce our new colleague, Dr. R.V. Hangarge, sir. Sir has completed his MSc and PhD from Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. Sir has also done his postdoctoral research at Department of Chemistry, Catholic University, Leuven, Belgium. Sir has published 25 national and international research papers and 4 patents to his credit. Sir is having H index 7 and I index also 7, and Sir is having 350 citations. Sir has 15 years of research experience in drug discovery in various multinational companies. Sir has received many awards and honors. Like in 1988, he has received the gold medal uh, at Dr. Babasaheb Ambedkar Marathwada University for receiving the first rank. Then in 1988, he also received 3 more prizes for standing first in the Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. Then in 2000, he has received CSIR SRF Research Fellowship. Then in 2001, he has qualified the SET exam, and 2002, he has done postdoctoral research fellowship at Belgium. So with this brief introduction, I request Dr. Hangarge sir to guide us. Over to you, sir.
Thank you, ma'am. Thank you for such a nice introduction. So I'm sharing my screen. Is it visible?
Yes, sir.
So, first of all, I would like to take an opportunity to thank our director of the institute, B.T. Jadhav sir, BOS chairman, V.B. Sawant ma'am, and head of the Department of Chemistry, BC sir, and my colleagues. So today we are going to discuss about the BSc Drug Chemistry FDP program. So in my this lecture, we'll discuss approaches towards drug and its synthesis. So the Sheikh has already discussed about drug discovery and development with drug. In my presentation, some points are getting repeated, but overall, it is a drug chemistry. The term, in short, the term drug means particles which are helpful in the diagnosis, cure, mitigation, treatment, prevention, and the disease control is used as a drug. There are different type of drugs. So some are bad drugs and some are good drugs. So which is a good drug and which is a bad drug, that we should know. So bad drug means what? Which is creating problems like we can say that addictiveness, that is the creation of problem because of drug. And what it means by good drug, which helps in the curing of the disease or in the diagnosis of the disease, that is called as a good drug. So this is the broad difference between good drug and bad drugs. So there are, again, drugs are classified into different categories. Some in the first image we can see here. In the first image, you can see that this is a local anesthesia used in a cancer type of case, cancer patients. Okay, this is the injection we everybody knew about it. Then this last image, that is a bronchodilator, means which is help in the asthmatic patients. So that is also one type of drug. and the addictiveness which is created because of this tobacco consumption. Tobacco is nothing but having a nicotine present over it. So that is the problem created in the due to nicotine, and that is a addictiveness. In the this polythene bag, you can see there is a some narcotics type of drug present in. Some are the good examples or good drugs, or some are the bad drugs. So what are the sources of the drugs? Where from we get the particular drugs in the market or from the shop? We know that the simple principle, we go to the medical shop and buy the drugs. But actually, what are the sources of such type of medical drugs? So first one is the animal source. That is the animal insulin, which was isolated from the pig and cow and sometimes human also. Some growth hormonal plant details, morphine. We know that the morphine is a narcotic drug. Inorganic arsenic mercury lithium, that is homeopathic drug nowadays used for the immunotherapy treatment. A synthetic chemical, that is the propranolol, and there are many drugs which are available in the market by which are prepared by the synthetic way. Biological drug, that is the penicillin, which is isolated by the serendipity, and the biotechnological drug, that is the human insulin. So in the image, you can see that I have shown here this honeybee, here is a frog and some plant, and this is the marine drugs. So these are the different drugs isolated from the natural sources. In the second slide, drug discovery and the development. How are the drugs discovered and the developed? The question is that we are, while taking the any pills of the drug, we easily take it, but never thought about that where from it come, how it came. We don't know about the background of that drug. So on that basis, what are the efforts chemists and the scientists have taken that we should be considered? New drugs are occasional new drugs found by accident. Those are called as serendipitous drug or serendipity. More frequently, they are developed as part of an organized effort to discover new ways to treat specific disease. Now that is called as a targeted therapies. What is meant by targeted therapies? Particular drug should help for the particular treatment, that is called as a targeted therapy. If you are someone is having headache and if you take a painkiller, then that should relieve the pain of head. Means headache should be decreased. That is called as a particular drug or target specific drug. But in some cases, what happens if you take a, combiflam or a, diclofenac, so whatever the pain, wherever the pain in your body gets stopped. That is called as a that's called as action of the drug or target specific drugs. So there is a long road to new medicines. There is a around 10 to 15 years are required for the invention of one drug to come into the market. Means from invention to the market, that requires around 10 to 15 years. Only exception is that in case of cancer, there is a short period, around 5 to 8 years, there is the chances of coming the drug in the market. So first one, where it will begin, there should be an idea in the mind. This is indicated by the bulb, where when that idea comes in the mind, the project team sit together and finalize that whether we have to go with this particular idea or not. There is a decision maker. They are the decision maker. Once they make the decision, then they will go for the synthesis in the laboratory, carry out the synthesis of particular entity or the compound in the laboratory, and found that once it is synthesized, then tested for activity. Means early testing. Early testing means what? In vitro testing. In vitro testing is nothing but the outside of the body. Means particular cells are or enzymes are isolated and treated outside the body. That is a petri dish. You can say that. Once it is showed good results, then move to the animal study. But means animals means what? Mice, rat, even guinea pig. Such animals are used. Even dog also used for the carrying out the study. Now, once candidate is selected, means animal showed a good response to the drug, then it moved to the candidate selection. Means we have to carry out around 10-15 drugs on the animal. Then one of the drug is getting selected for the further study. Then carry out the excessive safety study of particular drug, and then carry out the formulation of that drug. And then once it is proved the efficacy of the drug, efficacy means how much dose is required for the treatment of particular disease, and is it giving the good results or the bad results. This is the one thing. And the second thing is that the effect of drug on the body, is it good or bad, and the effect of the body on the drug, both study are carried out simultaneously. And once though that study is completed, then you have to carry out the study in the volunteers, healthy volunteers. Then the volunteers in the range of 3 to 5 only, that is called as phase 1 study. In the second phase study, 100 to 300 patients are there. In the third study, phase 3 study, there are 3 to 10,000 patients are there. This is about the antibacterial, antimicrobial studies. These are considered. Then the filing of NDA. Before that, we have to go for the clinical trial. NDA means new drug application, and at the last, registration, and the new drug comes in the market. That is a new medicine. This is the pathway required for the completion of from invention to the drug into the market. So what is this? Is this span is around 10 to 15 years. Except cancer drugs, the span is reduced to 5 to 8 years, because we know that cancer is a very dangerous disease. So to take early cure from that disease, the patient is started early for the treatment, and there is a unmet need of medicines in this particular therapeutics. So this drug discovery approaches, there are 4 drug discovery approaches. First one is the serendipity, means accidental results, whatever we are got, those are called as the serendipity. Then the second one is the screening study. Then third, chemical modification. And fourth one is the rational drug design. Means in the serendipity, almost 5.8%, 84 drugs out of 1437 of the drugs on the market are serendipitous. Serendipitous means accidental results we got that. That is called as serendipitous results. Of these drugs, 2.2% were discovered in the laboratories. While carrying out the experiment, actually the drug is made for different purpose. And while testing out that particular drug in the laboratory, it is found that this is useful for this use rather than the earlier, means previous one. So that is, there are 2.2% of such drugs are available in the market. 3.7% were discovered in clinical settings. Once that drug is given to the patient while carrying out the clinical studies, in that clinical studies, it is found that this drug is useful for particular use. Okay, so like this invention or serendipitous results are occurred. 18.3% pharmaceuticals in clinical use are derivatives of drugs that were discovered with the aid of serendipity. Suppose some drugs are there. Elliptician was the first drug was isolated from the kadipatta leaves. You know that. That is a carbazole alkaloid, and from that alkaloid, the scientists have developed different type of anti-cancer drugs over that. So this is the use of serendipitous results in the early years, and now also many drugs are coming like a serendipity. Penicillin is the one of the example of serendipity. Total to 24.1% of drugs can be addressed as a serendipitous. Okay, means there are total 24%, means you can say that 1/4 of the drugs are the serendipitous in the market, and non-serendipitous are the 75.8%, means around 76% of the drugs are the unserendipitous. And what is important here, screening studies. So drug discovery and the serendipity we have studied now. The antibiotic penicillin is the one example. Propranolol is another example. That is a beta blocker. And the third one is a tolbutamide. That is these are the 3 major and available in the market drugs are serendipitous. So random screening. What is meant by random screening? In the library of the compounds, we know that the identification of the scaffold first, means the structure identified first, and study of particular examples taking randomly and check it out whether this compound is useful for any therapeutic area like cancer, diabetes, cardiovascular diseases, pancreatic diseases, colorectal cancer diseases. Means whatever the diseases are there, you have to study against such diseases, and if it showed some promising activity, then you modify such compounds and make a useful compound out of it. So this funnel indicates that at the beginning, this green top of this funnel indicates that discovery and the screening of the compounds, then that narrow down to the lead optimization. And the third point comes, that is admit. Admit means what? Absorption, distribution, metabolism, excretion, and the toxicology of the compound. Then the that blue range of this funnel indicates that the clinical trials. Clinical trials are three clinical trials. Most three are the clinical trials: phase 1, phase 2, and the phase 3. Once it completed these three phases, then compound moves to the NDA approval, and that once it is completed, that application process, NDA approval process, then it moves to the new drug to the market. Means these are the many hurdles we have to cross, then only we reach to the new drug into the market. So just now I said that ellipticin is the compound which was isolated from the kari patta leaves. Then what is that exactly? So lead generation or the natural ligand screening, that ellipticin was checked against the anti-cancer and it showed some good activity. And after biological testing, it is useful for anti-cancer. Then carry out the modification around that structure and synthesize many compounds, and those showed the promising activity. Then take those compounds into the preclinical studies. That is called as a chemical modification in the compound. So you consider this example, which is most of the guys might have taken this drug. I expect everyone have taken this drug by themselves. The drug is a ranitidine. It is a proton pump inhibitor. We can say that target is a proton pump inhibitor, ranitidine. Actually, its first fragment, its first precursor is, we can say that is a cimetidine. Cimetidine is modified to the ranitidine. What changes they have made? If you look at the structure, this urea part actually, this urea part, here is a cyano is replaced with the nitro group. Both are electron withdrawing groups are over there. The remaining, this part, this is a side chain which is a fix. There is no change at all. This imidazole portion is removed with the furan having this side chain with the dimethylamine. Why we put here dimethyl? Why those guys put here dimethylamine? There is a reason, because this imidazole is having some basic character. That basic character should be come into the this ranitidine molecule, so that they put a dimethylamine, and which is easy to form the salt, and so that it is the absorption get increased. As Shakespeare said that the bioavailability of the compound get increased, so by making it salt or the product. Like that way, the conversion of cimetidine into the ranitidine, and ranitidine cost the billion dollar market in the US and the worldwide. So methodologies and strategies of CAD, computer assisted drug design. CAD means computer assisted drug design. There are four different ways, or major are the two different ways to design such type of strategies. First one is a direct design and the indirect design. What is meant by direct design? The direct design is a structure-based drug design. Here, structure is known. Structure of compound, that is not important here. Structure of the protein, that is important here. On the basis, or after knowing the structure of protein, then we can carry out the required ligand to attract that particular protein and show the good activity, and that is called as direct design. In the other way, that is an indirect design, that is a ligand based drug design. So whatever the protein is there, we don't know the crystal structure of particular protein. What you have to do, you have to take a ligand means here or organic compound or inorganic compound, whatever the compound you want to study, that compound you have to take and trial error basis. We have to take it whether it is fixed or not, or if it is fixed, whether it is showing the good activity or not. That you have to check. That is carried out with the help of computer assisted drug design. Nowadays, everybody is applying such principle in the drug discovery and the drug designing. So why it is important? Once we know that, means all faculty members once know such type of technologies are applied in the nowadays in the drug discovery department or in the drug chemistry. So we can tell the student such type of things, techniques are applicable in the finding of particular drug for particular treatment or particular therapeutic area. So what are the drug targets? So molecular structure with an organism linked to a particular disease whose activity can be modified by the drug. So every drug work for the every purpose? No. We know that if you are having headache, you have to take a particular tablet. If you are having a stomach, then you have to take a different particular tablet to show the good results. Okay. If someone is having different problem and someone taking the different medicine, it would not show the any good results. Okay. So in that case, what you have to do, first of all, you have to study the drug targets present in the human body. So 70 to 80% of the drug targets are enzymes in our body, human body. Around 80% targets, biological targets are enzymes. Then the second point comes that the receptors. Then the third one is the monoclonal antibodies. Fourth one factor, binding protein, transport protein. This is decreasing order. But the major one that we are focusing here, that is the enzyme. So enzyme is a playing important role in the therapeutic area that is there. Now distribution of target by biological criteria. So what are the roles of particular targets or distribution of such targets by biological criteria? Neoplastic diseases, hormonals and hormone antagonists, viral infections, blood and blood forming, means these are the some specific we can say target specific distributions of a biomedical criteria given. So 12.15% is involved in the viral infections. Nowadays we are facing bigger problem, that is the coronavirus and COVID-19 situation. This pandemic situation, we are facing lot many problems over here. So that is affecting 12.15% viral infections. Okay. So while we are focusing now the drug synthesis, while carrying out the drug synthesis, we have to consider 5 important parameters. What are those 5 important parameters? We can say in short, it is admit, not admit, it is admit. Admit, A means for absorption, D means distribution, metabolism, and uh, toxicology. These are the points to be studied or bring under consideration while carrying out the synthesis of particular drug. What is meant by absorption? Absorption is a phenomenon. Whatever the drug we are getting inside the body, where how much it is getting absorbed by the particular target, that is called as absorption. So whether that drug is whatever the drug is absorbed by the body, whether it is distributed to the each and every part where it is required or not, that you have to be determined. Then metabolism means excretion of that particular compound, whatever you have taken as drug should be excreted in the form of metabolites, and that metabolites should not be toxic. So that this admit property, toxicology study of particular drug is also studied. This admit property is very important, and once it's clear this admit properties, then we can move to the pharmacodynamic study, pharmacokinetics and the pharmacodynamics. This pharmacokinetics means the action of the body on the drug that is checked over there in the pharmacokinetics, and in the pharmacodynamics, action of the drug on the body that is checked in the pharmacodynamics. This is the major difference between PK and the PD. PK means pharmacokinetics, PD means pharmacodynamics. That is there. So we are taking one or two examples of a drug synthesis. We know the very well-known example, that is the paracetamol, which is synthesized from the phenol. By first of all, it is carrying out the nitration reaction. In the nitration reaction, we are getting two products. First one is the orthonitrophenol and the other one is the paranitrophenol. So here the required compound is a paranitrophenol, which can be separated by distillation or crystallization, whatever is their technique for the separation, we have to apply over there. Once we isolated the desired compound, that is the paranitrophenol, which on reduction with sodium borohydride gives the amine, and that on acetylation with acetic anhydride, that is give a paracetamol. But we know that reduction of nitro compound with sodium borohydride is very costlier, because as we know that sodium borohydride is an expensive reagent, and we cannot use such type of reagents in the bulk scale. So what you have to use, you can use either iron reduction, tin reduction, or such type of reductions we can carry out for the reduction of nitro to NH2. And once you have NH2, that is the second step is the acetylation reaction. That is nothing but a paracetamol molecule which is available in the tons in the market. Now the second one, aspirin. That is aspirin is the trend name, but the full form of that aspirin is acetyl salicylic acid. We know that salicylic acid is a 2-hydroxy benzoic acid. On acetylation, with the help of acetic anhydride, we get a acetyl salicylic acid. So this nowadays we are facing many big problem like what you call that is a coronavirus, and the market of these viral drugs are day by day getting increased. So the synthesis of gancillovir synthesis I have shown here. The synthesis was initiated using epichlorohydrin and benzyl alcohol. Here, the 2 moles of benzyl alcohols when react with the epichlorohydrin in basic condition, here base is used as sodium hydroxide. You give the base bis hydroxy compound. Okay, that hydroxy compound on reaction with the paraformaldehyde and hydrochloric acid, and there that OH get converted into the OCH2Cl, and that CH2Cl reacts with the potassium acetate to get converted into O acetate, and just O acetate react with the diacetylated product of purine in presence of paratoluene sulfonic acid, and which on reduction or the removal of that acetyl group will get a desired compound, that is a gancillovir as a target molecule, which is used for the viral treatment. Now, very important, I want to mention here role of catalyst in the drug synthesis. So, what is the role of the catalyst in the drug synthesis that we have to study? There are many compounds. We know that right hand and left hand is not same. We have seen in the stereochemistry or enantiomers of chiral objects that we have studied. What is important here? The particular use of particular catalyst for particular transformation. Means if you use R isomer is showing some biological good activity, whereas S showed some bad activity. So separation of these two these two enantiomers gives the beneficial effect. Sometimes synergistic effect also shown. We know the example of citrazine, levocitrazine, L-citrazine and R-citrazine. Here, levocitrazine is the most active than the other form of the citrazine. So that levocitrazine is costlier than the citrazine. We know that citrazine is having, earlier people were using citrazine, nowadays people are using a levocitrazine. This is the use of catalyst in the drug discovery. So sometimes what happens, you have to use the chiral catalyst. See, it is showed here R gossipol, S gossipol. This showed a difference in the activity, and see R binol and S binol. Means what is important here? If you use one compound like R isomer, then you will get either S or the R isomer as a product. On this demand, the catalyst playing a very important role in the drug synthesis. Now the therapeutic areas. So these are the therapeutic areas in which I have worked. So first one is the anti-infective, that is called also known as antimicrobial or antibiotics. So antibiotics are medicines that help stop infections caused by the bacteria, and they are target specific. So if someone is having infection like a skin infection, a urine infection, or urinary tract infection, some infections are having, so you have to use a specific medicines for particular use. Currently, what it indicates, you have to choose the particular drug to have a safe actions of that drug. So dosage is also important. You know that azithromycin, if doctor has prescribed to you, you have to take maximum 3 days. If you take more than 3 days, then you have to face the adverse effect of particular azithromycin. So different patients, there are different patients, means someone is having different infections, that is depends on that particular that use, you have to use different medicines. Drug action, you should not take two medicines at a time which may have some drug interaction, and due to that drug interaction sometimes severe adverse effects may get occurred. So to avoid that, you have to use drugs separately. Now there are top 10 class of antibiotics in the market. So penicillin, tetracycline, cephalosporin, quinolones, lincomycin, sulfonamide, glycopeptides, aminoglycoside, carbapenem, macrolides, ketolides, and oxazolides. These are the 10 major antibiotics present in the market. So if you go to the doctor, if doctor prescribed antibiotics, they will prescribe among these only, because they have captured big market, and they are having, or they have proved their efficacies and target specific activities and for diseases. So I have worked on the macrolides, ketolides, and oxazolidinones. So this is the patent which we have published, and it is granted in the US. Now the next one is the anti-diabetics. Anti-diabetic drug are all pharmacological agents that have been approved for hyperglycemic treatment in type 2 diabetes. So we know that diabetes is a nowadays it is a very dangerous disease. We can say that it is the mother of all diseases, we can say. From this, once this disease is started, the other all diseases comes around and roams around this one. We know that sugar is a very important. Okay, so diabetes, there are two types: type 1 diabetes and type 2 diabetes. Now we are focusing type 2 diabetes. Beta cells are playing very important role in the diabetes. We have studied for the secretion of insulin, but this drug, that is the SGLT2 inhibitors, are playing important role in the kidney. So earlier drugs were playing important role in the liver, pancreas, but this drug particularly playing important role in the kidneys. What is that? Insulin. Earlier drugs are insulin, preliminitide, GLP-1 receptor, and oral hypoglycemic tablets were there. Now that this is a new group for oral medication, work helping the kidney to lower blood glucose level. Means with the help of kidney, the recycle of reabsorption of the sugar get decreased. Okay, and this drug is approved in the year 2013, and they are taken once a day with or without food. That doesn't matter. Some these are the examples. That is the Foxica, dapagliflozin, canagliflozin, and impagliflozin. These are the examples of these SGLT2 inhibitors. So, what are the side effects of such drugs? You know that every drug is having some side effects, but this drug is having lesser side effects. And what are those side effects? Are urinary tract infection. Mostly it is occurs in the women's more. Hypoglycemia sometimes get occurred, but with the help of dapagliflozin, there is a no hypoglycemia get occurred. Diabetic ketoacidosis means decay. This is a one of the major drawback of particular this drug, that is a dapagliflozin or inokana or jadians, that is a decay diabetic drug. So this is the glomerular filtration. I'm not going to detail about this. This is a biological part. Okay, this is the SGLT2 inhibitors. Sodium and potassium influx outflux get occur, and the reabsorption of sugar takes place over there. Now the third one is the anti-cancer. Anti-cancer begins when genetic changes interfere with the orderly processes. What happens? This cancer begin when genetic changes. You listen carefully. Genetic changes means what? Hereditary changes one also comes in that picture. Uh, we know that breast cancer. Once the family background tells that someone is having breast cancer in your family, there is a chance to get a breast cancer in your family members. Okay, so if it is not there, and the what do you call it, invasion of particular that cells, breast cancer cells migrate to the many part of the body, so that to be taken care of it. So cancer is the uncontrolled growth of abnormal cells in the body, right? Cancer develops when the body's normal control mechanism gets stops working. That is there. Old cells do not die and they instead grow out of control, forming new abnormal cells. Means what indicates there is the uncontrolled growth of the cells get occurred. Second thing, there is the no dying of the cells and formation of the new cells are there, and the extra cells may form a mass of tissue together, and that becomes a tumor. Some cancers such as leukemia do not form tumors. Leukemia is a blood type of cancer, blood cancer. So these are the some cancers occurs there, and their percentage, their statistics are mentioned here. The in main, the prostate cancer is a 25%, and other comes are 20%, lung cancer 15%, bladder cancer 7%, colorectal cancer 10%. This is the percentage of the cancers getting over there. So what are the key facts? What we have to the precautions we have to take? So cancer is the second leading cause of the death globally. So 9.6 million deaths in the year 2018. Approximately 70% deaths in a low and middle class income families or income countries. So, what are the precautions we have to take? High body mass index, low fruit and vegetable intake, lack of physical activity, tobacco and alcohol consumption. These are the five major parameters playing important roles in the cancer. So what is important here? Tobacco use is responsible around 20%, 22% of cancer death, and hepatitis and HP, human papilloma virus infections, are responsible for up to 25% of the cancer. Only 26% of low-income countries reported having pathology services, and treatment comparison 90%, 30% of high-income, low-income countries. Total annual cost of cancer remedy in the year 2010 was 1.16 trillion, not billion, it is a trillion. We can say 1 million is equal to 10 lakh. Okay, so 1.16 trillion is the cost was in 2010, and now into 2020 it is almost 2.5 times. Only 1 in 5 low and middle income countries have the data to drive cancer therapy. So what is the background of the invention? So cancer cell possesses certain characteristics that allow them a growth advantages. In whenever a being is having the cancer, that cancer cell get a particular required environment to grow that those cancerous cells. These include six main alterations in the cell physiology. What are these cell physiologies? Are there self sufficiency in growth signals, okay, insensitivity to growth inhibitory signals, evasion of apoptosis, indefinite proliferative potential, sustained angiogenesis, tissue invasion, and metastasis. Means these are the some difficult words. So once we understand such words, meaning and concept, then we can easily teach to our student. So these are the drugs which are useful for this target, this therapeutic area, and these things. A large majority of cancers are known to be mutations in growth factor signaling cascades that lead to constitutive activation of these pathways. So activations in the pathways are very important. Now the invention relates to anti-cancer compounds. So look at this structure. This such type of structure first invented by GSK, and they bring their molecule in the market like a mechanist. Okay, and that mechanist compound is useful for the treatment of melanoma. So what is melanoma, that we are going to discuss in the further part. So anti-cancer compounds, pharmaceutically acceptable salts or the solvents, combinations with a suitable medicament and the pharmaceutical agents to improve or to get the synergistic effect. Methods of treating various cancers are there. Before knowing these methods, we should know biological target of particular compound. That particular target is very important. The target name is a MAKE. MAKE is having full form mitogen activated protein kinase KK pathway. So here GF is indicated here, that is a growth factor. This is a cell membrane. Across this cell membrane, this is the biological part. This activates GRAP2 protein. That activates SOS. That SOS activated this RAS protein, which is the inner side of the cell membrane, and that on a phosphorylated RAP, BRAP, and there is activation of MEK kinase, and once that MEK kinase activate ERK, and there is the cell division get occurs in the nucleus. To avoid such cell divisions, the GSK use a tremetinib compound, comitinib compound, and binimetinib compound for the control of these cancers. Now, what happens? This is the target. Okay, we have studied, but how this cancer get occurred? The cancer get occurred because of the mutation comes in the RAS, RAF. These are the two proteins wherein which the mutation get occurs. Once mutation get occurs, there is a chance of we can say resistance to the particular drug. So once resistance come, how this resistance come? This pathway was going downwards like this, but beyond that, there is another target, or another pathway is created by that enzyme to activate the signaling to the center. That is the resistance to make inhibitor get occurred, and this is the patent which is published in the year 2016, and it is granted patent in the US. MEK inhibitor. Heterocyclic compounds as a MEK. Okay. These are the MEK inhibitor which is coming in the market. This is in a phase 2 completed. It is in a phase 3 trial now. This is the invention of my compound. Now melanoma. Melanoma is nothing but a skin cancer. Is the deadliest of skin cancer. Rate of diagnosis for the disease have increased nowadays. So it looks like a mole on the body, and it spread like anything over all the entire part of the body. Today, it is one of the most common cancer found among young adults in the United States. So prevalence of melanoma, 1% of all skin cancer cases mostly occurs in the Western countries. In the 2020, there was estimation around 1 lakh people get suffered with this, and there is no treatment. There were no treatment available. Now it is treatment is available as a mechanism which is invented by GSK, and it is a solvent form. It is DMSO solid form, and this compound is having some adverse effect. And these adverse effects, we were trying to improve the adverse effect, and we got the success in that compound. What are the adverse effect of these compounds? Adverse effect means a side effect, we can say. Increased alkaline phosphatase, dermatitis infection, stomatitis, high blood pressure, abdominal pain, hemorrhage, dry skin, itching, and the major one is the ocular toxicity. So ocular toxicity means ocular means eye toxicity. This ocular toxicity can be removed with the help of our drug which is invented. Now around 4 scaffolds we focused on this. These are the scaffolds. Actually, why I've taken this example, because this coming into the market, I know the beginning from this designing to the market, that we have known. Around 1,000 compounds were synthesized in this series from these 4 scaffolds, and this around 1,000 compounds, only one entered into the phase 3. Okay, so in vitro biological data suggests that IC50 values. What is meant by IC50 values? Inhibitory concentration 50%. That is 1 nanomolar to 400, and means below 500 nanomolar. 58 compounds showed these results. In the B group, IC50 is 0.5 micromolar to 1.5 micromolar. Only 13 compounds showed, and IC50 1.6 micromolar to 3 micromolar, 6 compounds. Okay, so what is important here? Whatever the IC50 towards lower side means 1 nanomolar to 500 nanomolar, that is very important for us and playing important role for our invention. Now there are many compounds. See here, compound number 2, 5, 16, 37, 19, 18. These compounds showed a good results. The percent inhibition of MEK phosphorylation at concentration of 100 nanomolar, 10 nanomolar, and 1 nanomolar, it is showed here. This data indicates, see this percent phosphorylation. We know that MEK activation pathways are getting activated with the help of phosphorylating reagent, which can be stopped with the help of inhibition of this phosphorylation, and this is carried out by these compounds. Okay, compound number 2, compound number 5, 9, and 35 were taken for the further study, that is in vivo biological data, and this study was carried on the nude mice on the 15 days at the concentration of 0.2 ml PBS. PBS means a protein bovine serum. A post-cell inoculation tumor dimension were measured when tumor becomes palpable. The tumor volume of that tumor we grown in the mice is measured with the calculation length width into width divided by 2. It gives the tumor volume in millimeter. And that study was carried out 21 to 22 days. Okay. And compound number 25935 were tested for tumor growth in colorectal cancer 205 xenograft model, and and that came in the phase 3 compound. Okay, So useful for the treatment. This compound is useful for the colon cancer, pancreatic cancer, lung cancer, melanoma cancer, papillary thyroid cancer, and non-small lung cancer cell. Okay, so this compound is in phase 2B clinical trial, having improved in a toxicity, easy to synthesize, because it is not having any chiral center. Scalable route is optimized and executed because this compound, uh, once it is identified as a target oriented space therapeutic compound, then need to be carried out in a large synthesis. So it is not having any chiral center, so that this synthesis is very easy. Route is optimized for the large scale, around 50 kilo scale. We have tried. Progression free survival is increased by 6 months only. And important thing is that this compound is useful for the pancreatic cancer. Earlier, there were no drug for the pancreatic cancer. They are having BRAF mutation and the KRAS mutation in that enzyme. So this patent is granted in all countries. This is about my presentation. Before ending my presentation, I will go through this melanoma slide. See, this melanoma is a very dangerous disease, skin disease, and that is causing many people are getting affected with this disease worldwide, mostly in the Western countries. Such disease is not present in India. Keep in mind. Okay, don't worry about this. So that compound was out licensed to Boringel England with the sign of $700 million deal for anti-cancer drug, and these are the paper notes which I have taken. Out licensed to BI, that is a 5,000 crore rupees, and this is the second partnership with this German drug maker Boehringer Ingelheim. Thank you very much. Say no to the drugs. This is my slogan.
Thank you, sir, for such a nice information. Now I request Dr. P.A. Bharad ma'am to express vote of thanks.
Thank you so much, sir, for detailed presentation. As we are very new to this area, so we are having so many questions. So I think that we will discuss later on in detail. So as they discussed methodologies and strategies of CAD, then drug synthesis, absorption, distribution, metabolism, excretion, and synthesis of paracetamol and aspirin, and many other things you have already discussed. सो फक्त मला असा प्रश्न विचारायचा आहे की आपण आता मुलांना शिकवायला सुरू करताना जसं तुम्ही सांगितलं की ड्रग काय आहे, बॅड ड्रग, गुड ड्रग, असं सगळं ते झालं. फक्त म्हणजे एक त्यांना आपल्याला म्हणजे असं अट्रॅक्ट करून घेण्यासाठी अजून म्हणजे आपल्या पहिल्या 2-3 लेक्चरमध्ये असं काय वेगळं सांगता येईल त्यांना आपल्याला, ते थोडंसं तुम्ही इलॅबोरेट करा.
बर, सुरुवातीला मॅडम आपल्याला काय करता येईल त्यांना, की ऑपॉर्च्युनिटीज काय आहेत. आजकाल मुलं जे आहेत ते जॉब ओरिएंटेड बघतात. मला जॉब कसा मिळेल आणि माझं सर्वायवल कसं होईल, हे याच्यावर मुलांचं लक्ष असतं. तर त्याच्यासाठी आपल्याला आधी काय करावं लागेल, की त्यांच्या ऑपॉर्च्युनिटीज त्यांना कुठे कुठे आहेत हे त्यांना एक्सप्लेन करणं गरजेचं आहे. आपण जर त्या बाबतीत बोलायचं झालं तर इंडस्ट्रियल ऍप्लिकेशन्स त्यांना सांगायचे आहेत. त्यांना घरातली उदाहरणं देऊन सांगायची आहेत, किंवा आजूबाजूची उदाहरणं देऊन सांगायचे आहेत, किंवा रिसर्च मध्ये करिअर करता येतं हे पण त्यांना पटवून द्यायचंय. एखाद्या सायंटिस्टनं इन्व्हेन्शन काय केलेलं असेल ते त्यांच्या समोर मांडणं गरजेचं. ह्या जे 4-5 गोष्टी त्यांच्या समोर केल्या गेल्या तर ती मुलं डेफिनेटली अट्रॅक्ट होतील. आणि त्याच्या पलीकडे जाऊन ड्रग केमिस्ट्री हा एक आपलंच कॉलेज, म्हणजे आपलंच एक इन्स्टिट्यूट असेल की जिथं अंडर ग्रॅज्युएट लेवलला सुरू होते, मास्टर लेवलला फक्त नगर कॉलेजला आहे, त्याच्या व्यतिरिक्त दुसरीकडे कुठे नाही. म्हणजे त्यांना इथं बॅचलर करून, अंडर ग्रॅज्युएट करून पीजी साठी त्यांना तिकडे जाता येईल, किंवा आपल्याकडे पण पीजीचा डिप्लोमा, पीजी जर सुरू झालं तर त्यांच्यासाठी बेनिफिशियल होईल. आणि रिसर्च करण्यासाठी मल्टीनॅशनल कंपन्याज जे आहेत उदाहरणार्थ झालं जर्मन बेस्ड कंपन्या BASF असेल किंवा GVK असेल, बायोकॉन असेल, सिंजेंटा असेल, ह्या BASF ह्या सगळ्या कंपन्या आहेत. इंडियन तर आहेतच आहेत, या व्यतिरिक्त मल्टीनॅशनल कंपन्या सुद्धा भारतामध्ये येऊ घातलेल्या आहेत आणि त्यांच्यासाठी आपल्याला मुलांनाही आपण सांगू शकतो की जॉब ऑपॉर्च्युनिटीज भरपूर आहेत.
Thank you so much, sir. Thank you so much for your detailed presentation. It will really help us when we will be teaching to our students drug chemistry subject, and we will need your guidance throughout this course. So thanks once again. I also thank our principal, Dr. B.T. Jadhav sir, and our head department of chemistry, Mr. P.V. Bise sir, for always supporting us. BOS chairman, Department of Chemistry, Dr. V.V. Sawant ma'am, for joining this session, and all my senior colleagues and all my other colleagues for joining this session. Thank you one and all. So with the permission of this organizer, I declare that the session first is over. So we will have break up to 2 p.m. Then at 2 p.m. we will have the second session. So thank you all.
Thank you.
Thank you, ma'am.